All right. So we're continuing on innate immune response, right? We talked about inflammation already and phagocytosis. Those are really paramount defenses, but those are internal defenses, right? But we have first-line defenses that we're going to talk about today that help prevent the organisms from even getting in, okay? So although we talk about the immune system in several parts, we talk about innate, the components that you're born with, these first-line defenses that we'll talk about today, the ability to have inflammation and phagocytosis. You can even do these processes in utero when you're a fetus growing in your mom. The other component of your immune system is your adaptive. And this isn't able to, as the name implies, adapt and change as we go over time. But the two of these you'll see are linked together, right? You can't have one without the other. There are components of the innate that then help out the adaptive, do this ability of being able to adapt, change, respond to changing antigens that are out there. That's another term that we've used a lot, right? Antigen, and we will continue to use again. Its main meaning was antibody generators, right? But as we've already seen, that's not the only response that our immune system does, but it was one of the earliest that we were able to easily detect, okay? But for yourselves, always think of antigen as something foreign, right, to our body. It's a foreign substance. So it could be any number of things. It could be a bacteria, it could be a virus, it could be a foreign protein. It could be food, could be pollen, it could be any number of things. Antibodies are a product of our adaptive immune response. They themselves are proteins. And they bind to specific antigens, and this is the important part. They have specificity to them. So when we get to adaptive, we'll talk about those different components. But we're going to focus on just the beginning, as Carol alluded to, she's getting ahead looking at complement, which is great. I don't know that we're going to get to that today because I want to check in with case studies today. So I'm going to actually cut this lecture shorter than I normally would. So we're definitely, these are all your components of your adaptive immune system, right? We'll talk about the first line defenses, really important, your anatomical barriers. We're going to look at sensory systems, and one of your sensory systems is your complement. Your body innately has this ability to recognize certain patterns, right? And see this as a danger signal, set off the motions of things like inflammation, right? And having those phagocytic cells into the area. Cytokines are chemical messages. You could almost think of them as the hormones of the immune system, right? Just like our hormones carry the signal for our endocrine system, cytokines is the general name given to the chemical signals that our immune system uses to communicate. And we're going to look at some general categories. Thankfully, we're not going to memorize individual molecules and what cells they come from and which cells they target. We're going to keep this at a pretty basic level, right? This isn't an immunology course, right? But you do need to understand their importance and their existence. We've already looked at inflammation, and we've already seen how important that is to get the white blood cells to the area. And fever as a normal response, right? An innate response to infection sometimes. And we'll talk about the components involved in that. But as I said, we're just probably going to get through the first couple today. So this is an excellent diagram, right, showing you those different tiers that I've mentioned in a little bit different way. So we have our first line defenses, the blocking gates, right, stopping stuff from getting in. Our skin is the best protective barrier from stuff getting in. Very few organisms can penetrate it alone. Most of the time you have to have some type of wound in order for them to gain entry. Other surfaces that are open are bathed in some type of mucus. This is a sticky substance, right, that helps trap them, and it also can be moved. That mucus can be moved to move the organisms out of the body, right? Even within that mucus, there are certain substances that help protect us. Our skin has certain substances and environments that help protect us. And then if they get past, right, they get past the skin, they get past the mucus layers, then we have to be able to detect that they've entered the body, right? And this is where our sensor systems come into play, right? It's like, the barrier has been breached, right? So pattern receptors on the surfaces as well as inside of our cells, phagosomes and sentinel cells. So we have receptors, right, on our cells that have the ability to sense that foreign substances have gotten in, antigens have gotten in. These receptors are even inside of our cells, so even inside the cytoplasm of many different cell types. And then the complement system, which is a protein system in our blood and in our tissues, fluids, the fluids that bathe our tissues. This system, notice, can activate the inflammatory response. So can our cells activate the inflammatory response. And so when stuff gets inside the cells, and even cells themselves from the outside or inside can detect and elicit that inflammation response. Because what is that? What is inflammation at the main level?
It's the signal to tell people we need to fight over here.
We need to fight. We need white blood cells that can fight to come here, right? Think of it as an influx of white blood cells. Even though we talk about, right, the increased blood flow, the whole purpose of that is the white blood cells getting there, right, to fight, right? We may have fluid leaking out, we may have swelling, we may have pain, right? But those are all just outward signs of the main reason why this happens, and that is to get the white blood cells there and contain, trap, destroy invaders. Now, interferon response, we're going to talk about a little bit later. This has to do with viruses, okay? This is our defense against viruses. So it makes sense that we could set off this response when stuff gets inside of our cells. We'll talk about opsonization and membrane attack complexes when we get to complement, okay? But these are direct ways of tagging for destruction and destroying cells that this protein detection system can do, this complement system. So our first line defenses include the physical barriers we just talked about, right? So our skin, the mucus. But there are antimicrobial substances associated with some of these areas of our body, and we have normal microbiota, actually normal microorganisms living on and within our bodies, and them being there helps protect us. So our skin, as I said, very difficult to penetrate. The dermis is very tightly woven, fibrous connective tissue, right? So this is a very sealed up wall to help protect us. The epidermis is many layers of epithelial cells. The very outer layer of our skin is actually dead, right? These cells are being keratinized. This is a protein. And this helps make it, our skin mostly water resistant, repel water. And it also maintains a relatively dry environment. And as you know, all organisms need a certain level of moisture in order to grow, right? So by not having too much moisture available, it helps prevent growth. That layer of skin is continually sloughed off. So any organisms that you may have touched, right, and they attach to those dead cells, when those cells leave, they also a lot of times get sloughed off. The really gross thing is that a large percentage of the dust in your house is dead epithelial cells. And that's actually what dust mites are eating, is your epithelial cells. Sorry, but it's the truth of it. Okay, so when we look at it a little bit deeper, the transients, as I said, right, that are just catching a free ride off of you are what are landing on that epithelial layer, the epidermis. The dermis, the much thicker layer, is where your normal flora exists. And some of these are going to grow in, although not very common, in the sweat glands, again, because of the salty environment associated with sweat. But the oil glands, the sebaceous glands, and the sebum, that oily substance that you produce, associate with your hair follicles. This is so that the hair slides out of the follicle. That's why the oil is produced. But microorganisms can eat this substance. And sometimes you'll end up with an infection of the hair follicle. We call these zits, right, or pimples. This is when you have an overgrowth of the organisms. You may even have pathogenic organisms growing there, right? And you get the pus that will come to the surface. That's an abnormal situation, of course. And there's lots of factors that come into play why an infection of a hair follicle will happen. So where you don't have skin, you usually have mucus, right? We refer to these as mucous membranes. Your entire digestive tract, your respiratory tract, your urogenital tracts are bathed in mucus. And in fact, your digestive tract is actually considered external to your body, right? It is a tube that goes right straight through your body. It's actually considered an external environment, not an internal environment. So a tube right straight through your body. So this is a rudimentary drawing showing you those different areas, denoting the different areas of the mucus. With your intestines, the movement of stuff through the system helps protect you, right? That peristalsis, the muscular movement of moving stuff through the system helps protect you. Less likely of stuff to get in if it's continually moving through the system. And then you have, in association with the mucus that's produced in your respiratory tract, you have ciliated cells. These beat and move the mucus up, right? You can spit it out. Please don't do that, right? If you do, dispose of it properly because it could contain pathogens, right? That's the whole purpose of what spitters drive me crazy, right? It is the grossest thing. So you're actually designed to swallow that, right? And it's meant to go down your digestive tract and then out the body that way. We refer to this system as the mucociliary, right? Mucus ciliary escalator, right? Because it's moving the stuff up and out.
Unless you're a smoker.
Yeah, and if you're a smoker, you tend to damage those ciliated cells, which is not a good thing, which predisposes yourself to lung infections.
So it actually helps us to swallow it?
Yeah, it's okay to swallow it. It is actually okay to swallow and not spit out of your body. Because you're exposing others when you spit it out of your body, right?
Those aren't going to make it past the stomach acid anyway.
And that's one of the things we're going to talk about. So the skin and the mucous membrane. So the skin, as we said, from perspiration tends to be relatively salty. This salt is an inhibitor of microbial growth. As we know, it can cause plasmolysis, can suck the water out of them. Lysozyme is an enzyme found in a lot of our mucus that we produce. It degrades peptidoglycan. Remember, who has peptidoglycan? Gram-positive bacteria. So do gram-negative, but gram-positive are most susceptible to this because they're probably their outermost covering is made out of peptidoglycan. So this is an enzyme that literally goes in and breaks the sugars apart. Right? It can literally digest away their cell wall layer. Peroxidase enzymes break down hydrogen peroxide, but into toxic forms of oxygen, right? So this can be very damaging. And again, we're going to do this in a protective way. So that notice that lysozyme is in our tears, right? In other secretions, it's even within our phagocytic cells themselves. In our saliva, we have the lysozyme, we have the peroxidase, we have also lactoferrin. Lactoferrin is one of the proteins. There's several proteins out there that we have. Transferrin is another one you'll sometimes hear about. This has the ability to bind to iron. The reason for this in these substances is to sequester it away from microorganisms because they need it for their metabolism, just like we need it for our red blood cells. They need it for their metabolism too. Transferrin in our blood is mostly meant to help transport the iron because iron isn't a very soluble, water-soluble element. But in essence, too, it helps sequester it, keep it away from the microorganisms. And then we have defensins. These are protein molecules that can literally pierce the membranes of microbes and help lyse them. So notice on our skin we have normal microbiota. We'll talk about those guys in a moment. And the digestive tract itself is a very harsh environment, right? You produce hydrochloric acid. The main reason for that is to digest proteins in your stomach. This is a very low pH. This is very damaging to most bacteria. Very few of them can survive in this type of environment. So swallowing your spit goes to the stomach, right? And those organisms should be digested right along with your food, okay? And then there's a sharp change in the pH to a more neutral environment by the addition of alkaline substances from your pancreas, which drops the pH to a level where your other enzymes can work for the rest of your digestion and absorption that happens in your small intestine. Your large intestine is where your normal microbiota are existing, right? So there's about 1 billion bacteria per gram of feces that you produce, okay? These are the good guys, though, and these guys are basically eating what you didn't eat, okay? And some of them actually produce vitamins for you that you can absorb. Your reproductive tract is, especially for women, again, bathed in mucus, and there is a certain pH balance that's maintained there, an acidic pH again, so that most microbial growth is inhibited, but there are some normal microorganisms that can grow. Yeasts are one of them, as well as acid-producing bacteria, which is why it tends to be an acidic type of environment. Your urinary tract, one of your best defenses is drink lots of water and pee often, because that flushing ability of continually flushing stuff out stops stuff from being able to take up residency and grow. Your urine is typically sterile unless you have a urinary tract infection. And the dangers with urinary tract infections is that they could spread from the bladder up the ureters to the kidneys, right? And you definitely don't want that to happen. So how do these normal microorganisms that exist in our skin, in our large intestines, and for women in the vagina, how do they protect us? One of the ways that they actually protect us is what we refer to as competitive exclusion of pathogens. So pathogens are disease causers, right? That cause disease, are known to cause disease. If these normal microbiota are in that spot, they're like taking up the parking spot, right? It's like getting here at the beginning of the semester. Good luck trying to find a parking spot, right? So you got to come early, right? So these guys came early. The normal microbiota got that spot first. So because that spot is taken, there's no sites for the bad guys to bind to, right? The spot is taken. And they're going to consume the nutrients that are there. So not only can you not find a spot, you can't have breakfast or lunch because you can't park your car and get out and get to the cafeteria. Same thing with these guys, right? Even if they could squeeze in and find a place to park, right, they're now going to have to compete for food, right? Because everyone else got in line before them. So some of them actually even also produce toxic substances. So Propionibacterium degrade the lipids and they produce fatty acids. These fatty acids are toxic to a lot of different organisms, right? So they're byproducts. So by them being there, one taking up the spot, eating the food, and then their byproducts they're producing, right, help keep away the pathogens, the other microorganisms that would cause harm. E. coli even synthesizes special substances known as colikines. These, of course, are named after E. coli, right? And so these, again, are substances that we found that they produce that inhibit other microorganisms from growing in our intestines. E. coli is Escherichia is one of the main bacteria in your intestines, one of the largest populations. For women, the lactobacillus, as I said, that exist in the vagina, they're the ones that are producing acid substances that lead to the low pH in that environment, which is also antimicrobial in action, helps others from growing. Now, when we have a disruption of that normal ecosystem that's going on, whether it be in your skin or your intestines or for women in the vagina, then we can end up with infections either by pathogens or even by some of our own resident microbiota that are present there. In the case of Clostridium difficile, this is a bacteria. It's unfortunately a spore former that's present in the environment. The good news is most of the time, even if you ingest these spores, they're not able to germinate in the intestines because, again, they're not able to access the food sources because of the other organisms there and the competition happening. But sometimes when people go through intensive antibiotic treatment, remember antibiotics target just bacteria in general, just gram-positive or gram-negative. It doesn't discriminate between the good guys and the bad guys. So your normal microbiota are destroyed also in the process sometimes. And that's one of the main contributors to this type of infection is that the normal E. coli and other organisms that are inhabiting the gut get killed off by antibiotic treatment. And then the difficile, which is in the environment, is able to germinate in the intestines and cause a really bad infection. The problem with this guy is that he produces toxins that literally can eat your intestines. Okay. I've also been told by most nurses that this one smells really bad. I've been told that before you even walk into that patient's room, you know exactly what's wrong with them.
Nothing's supposed to smell that bad.
Yeah, nothing's supposed to smell that bad. Thankfully, I should hopefully never experience that. That's the fun you guys have to look forward, and some of you guys already have. So I learn all these cool things from my students sometimes too. So last semester, one of my students, or a semester before, I've lost track, one of the ways that they treat this is I actually do a fecal transplant. They give them someone else's feces to reestablish that microbial environment, which doesn't necessarily surprise me because the same thing happens with young animals, especially horses and cattle. You'll notice that the babies will eat their mom's poo, and one of the main reasons for that is to establish that microbial environment in their intestines, and it really is the microbes that digest their food for them. Cows, if you give cows antibiotics, most farmers will freak out because they're worried that it's going to disrupt their normal microbiota in their intestines that allow them to even live, right? If those bacteria die off, they will die as well. You had a question?
Yeah, weren't you saying earlier in the semester about the appendix not being a vestigial thing, that it actually repopulates the microbiota after some kind of infection or whatever?
I didn't say that, but I
heard that.
Yeah, it's probably possible, because the appendix, right, is the appendix in your cecum. Your cecum is where there's a lot of bacteria and fermentation and stuff can happen, but same is true for your large intestine. So it could be a source of repopulating if a large number of microorganisms get removed from the larger part of the large intestines.
I think that's what I was told that's
Yeah.
what they believe its actual purpose is now.
Yeah, now. In the
Before,
past,
yeah.
When we
Just like
ate more
tonsils and
roughage, it
everything
helped
else.
digest that kind of stuff. We don't actually digest roughage fiber, cellulose. It just goes through our system. We don't have the bacteria, we don't have that special portion in our intestines to be able to do the digestion like cows and horses and stuff do. So it could be it could be serving the purpose of maintaining our microbial populations for us. It definitely could be that.
How much of a positive effect can like probiotic substances and stuff like that?
Right.
Yogurts, stuff like
Right,
that?
so yogurts and stuff like that, a lot of them always had microorganisms in them that can be helpful, as long as they have active cultures in them and they haven't killed them off by pasteurization and stuff. So there is a lot of help to that, especially where Americans don't eat the best diets. Typically, if you ate a well-rounded diet, then you shouldn't have to worry about it. And babies in general, like when we're first born, we pick up the microorganisms from the environment necessary to populate our gut. And then the last one is Candida. Yeah, I mean, little kids put everything in their mouth. So they definitely populate very soon. This is a yeast that is part of the normal flora for most people, even guys. Women are the ones that unfortunately, because of the vagina, because of that organ, tend to experience an overgrowth of this yeast. And this, again, commonly happens after there's been a disruption to the normal ecosystem in the vagina. And most common thing that causes that type of disruption is antibiotics. But there are other things. Diabetics patients sometimes deal with it a lot because of the high sugar content in their blood. That also can end up in their urine, and it can really promote the growth of these organisms. They love sugar. Who doesn't, right? And the bacteria usually compete with the yeast for the food supply, and so we're able to keep a normal balance. So when we say a yeast infection, what we're referring to is actually a yeast overgrowth, right? Where the yeast are growing to such large numbers because they're not being kept in check by the bacteria. Hormone changes. Lots of things can affect your diet, right? Lots of things can affect. The most common, though, is antibiotics, right? Because they directly target bacteria. And unfortunately, they don't go after just the bad guys when you have an infection. They're going to disrupt your normal flora as well. Most of the time, we're able to easily reestablish this. So we've mentioned some of these guys previously, right? I introduced them. All right. And so we're going to look into the cells a little bit more in depth. We categorize them, and previously this objective just had granulocytes and mononuclear phagocytes and dendritic cells and lymphocytes. And I added in the actual cells that fall into these groups. So within our granulocytes, we have the neutrophils, the eosinophils, and the basophils. And remember I said, they all end in "phil," right? They're all filled with granules, which is why they have the name granulocytes. And it's also why their different names have to do with how their granules, how those vesicles stained when scientists first discovered them. Mononuclear phagocytes, also your granulocytes have that multi-lobe nucleus, where the phagocytes have a pretty uniform mononucleus, right? And then our lymphocytes all look the same under the microscope, unless you're using special dyes and instruments to look at them. And those are given the subnames of B cells, T cells, and natural killer cells, which of course is the coolest name. So these cells are always found, your white blood cells are always found in normal blood, right? They're always readily available. They're always traveling to where they need to go. Some of them will play dual roles in both innate and adaptive immunity, right? So they're your first line defense, and then they're going to help, right, in this adaptive being able to specifically target specific pathogens. So hematopoiesis refers to the formation of all your blood cells, even your red blood cells, happens in the bone marrow. And this happens from stem cells that are in your bone marrow. And this is why, we can do bone marrow stem cell transplants for people and repopulate these cells for them. Because those cells can become any one of the cells needed. How does the body determine which cells to make when and to what amounts? It's chemical signaling. So the chemical signaling group that's responsible are your colony stimulating factors. These are cytokines. These are chemical messages, specifically targeting the stem cells and telling them which group of white blood cells do we need. Do we need lymphocytes? Do we need phagocytes? Do we need granulocytes? Do we even need specifically down to neutrophils, right? These chemical signals are going to stimulate those cells to produce the population of cells that we need at that time and the numbers that are requesting. So it's like placing an order, right? Colony, because it's going to be usually a group of cells. So red blood cells, aka erythrocytes, right? These stem from your bone marrow as well. They carry oxygen in the blood. They play no part in our immune system, right? So that's all we're going to say about them. Platelets are actually, I think of them little bags of enzymes. They're not actually cells. They are derived from cells, megakaryocytes. These are very large cells that break up into these smaller pieces. They contain the enzymes necessary to start the clotting cascade, right? They can also be used as little sandbags, you could say, as patching up holes in your blood vessels. So they're very important to our blood clotting mechanism. And we're not going to go through all the particulars and stuff of blood clotting. We're going to leave that for A&P classes, right? They usually do a pretty good job of that. We're going to stick to the cells. So another name for white blood cells is leukocytes, right? I tend to stay away from that one, mostly because people in general sometimes confuse them with lymphocytes, right? Leukocyte is a general term for all white blood cells. I typically just say white blood cells. Pretty straightforward, right? When someone says lymphocyte, they're talking about a subgroup of white blood cells, right?
That's specifically in the lymphatic system.
Yeah, they're mostly in your lymphatic system. That's why they're named lymphocytes, yes. That's where they get that name. So these are our four major categories of cells. We have our granulocytes, right, which are their neutrophils, all the phils, right, neutrophils, basophils, eosinophils. The mononuclear phagocytes, we're talking about monocytes and macrophages. Dendritic cells are their own little group. They're so awesome. Lymphocytes are your B cells, your T cells, which are antigen specific. And then your natural killer cells, as the name implies, are naturally good at killing. So, as I said, it all starts out in the bone marrow, right? And then depending on which signaling they get, right, they'll differentiate into the common cells that give rise to the lymphoid or your lymphocytes, right? The good news, you don't need to know these guys, right? Okay? But know that it's a stepwise process of developing these cells. And there is actually a belief that monocytes, so this is what you would find in your blood, this line right here, okay? You'd see your eosinophils, you'd see your basophils, you'd see your neutrophils, you'd see your monocytes, you'd see natural killer cells, you'd see lymphocytes, and notice they look pretty much the same. They got them colored differently here, but they're not different. if you were to look at them under the microscope. These guys you would find in your blood. Your monocytes differentiate into macrophage in your tissues. Same thing with your dendritic cells, right? You're not going to find these guys in your blood. These are in your tissues. And these are subsets of your monocytes. So they have differentiated, right? You can almost say matured. They've changed in some way to function in your tissues.
Aren't mast cells found in the tissues too?
Yeah, and that's why they're on this line too, right? So also found in your tissues are mast cells. These guys are close cousins to the basophil, and that a lot of the granules, the substances they contain, are very similar to basophils. Okay? So yeah, that's the differentiation here, right? This is tissue line, this is blood line, right? What you would find in your blood. So granulocytes, neutrophils, these guys are responsible for the main phagocytosis, right? They're going to be first on the scene during inflammation, and they're only going to leave the blood and go into your tissue when they're being called there during inflammation. It's the only time they're going to go in. Basophils are involved in allergic reactions, so we'll definitely talk about them when we get to that. As well as the inflammatory response. As I said, they're similar to mast cells. The difference is that mast cells are found in your tissues. And as we saw before, right, these are the ones that are going to release things like histamine and elicit that inflammatory response. Eosinophils, they have been found that their granules are really good at fighting parasitic worm infections, and that they're also involved in certain allergy reactions. And again, this has to do with the particular substances they have in their granules. Right?
Don't basophils increase under a viral attack too?
I don't know.
I just wonder this way with the blood. We could screen it and tell. With the CBC with diff.
I'm not sure. I have to look that one up.
That's a broad idea, a basic idea of what because the neutrophils were sky high.
Yeah.
In the diff, we knew it was a bacterial infection.
Yeah, usually, yeah. So what she's talking about is that doctors a lot of times will order a test referred to as a differential, or diff for short. And what they do is they take a sample of the blood, and they're going to count up to 100 white blood cells. And that'll give you a percentage of these different types that you see in the blood. And when you see neutrophils, right, you're larger normal number than normal levels, because like I said, you always have a certain normal level, but these numbers are going to increase when we have inflammation, right? You're going to call them up. So particularly with bacterial infections, you'll get a really large number of neutrophils. So when that number's up, that's a kind of key that we might have a bacterial infection. When eosinophils go up, right, it could be a parasitic worm infection.
The basophils would be higher and the neutrophils would be more, and neutrophils and eosinophils would be more in the normal range.
Yeah.
Basophils would rise because of the inflammation process involved in a viral
Infection as opposed to bacterial.
But if it was only the basophils up, we knew we're looking at a virus probably.
Right.
So treat symptoms and run the course.
Right, yeah, right. So we're not going to go into those specifics for this course, right? We're going to keep it general. But know that, right, because of the different jobs of these cells, you know, they've worked out, if you look at the particular numbers, you can kind of get an idea of what type of infection is going on because it's based on the jobs of these cells, right? And it's been a while since I've looked at a lot of that. So, and we don't go to that level in this course, so I'm not going to say for sure.
Actually being out there doing stuff.
Right, yeah, but now you understand why, right? You understand why you see those increase in the certain numbers, right? So for our mononuclear phagocytes, so these are your monocytes that are circulating in your blood, okay? Then when they leave the blood, they're going to differentiate into macrophages and dendritic cells. Now your macrophages, all your organs, get populated by macrophages, right? These are your guys on-site, right, resident macrophages. Because of this, when scientists were first looking at organs and looking at tissues, they saw these cells. And of course, they named them. So for instance, in your liver, it happened to be the Kupffer cells. We'll come to find out it's just a macrophage that's living in your liver. That's it. Same thing with your mesangial cells in your kidneys. Thankfully, by the time they got to other organs, right, like your lungs, they're like, oh, it's just an alveolar macrophage. Just happened to be a macrophage that lives in the lung, right? We're just denoting it by the fact that its home is this organ, okay? But others will be called up during inflammation, right? These guys will be able to sense when there's invasion, right? They can release cytokines, and they'll call even more macrophages to the area. Dendritic cells, I nickname these guys the scouts because they're traveling around, right? They're found in your tissues, and they don't stay there, right? They're going to always antigen sample, and then they're going to bring this information. These guys are the major link between your adaptive and your innate immunity because they're going to grab up the antigens and they're going to go present them to your lymphocytes, right, that are, as he said, in your lymphoid system, right? Right, yeah. Do you recognize this guy? Do you recognize this guy? Right? They're trying to find that specific T cell that recognizes the antigen they found. All right. So, as I said, often sometimes named after in different organs. But note that it doesn't change the fact that they are a macrophage, right? They're going to engulf by phagocytosis and destroy. Dendritic cells are going to engulf, but that's not their main job. Their main job is to go and present those antigens. And we'll look at that specific interaction later. So sentinels, scouts, right? They're gathering information and bringing it back to base. They're going to engulf materials in the tissues, right? And as I said, they're going to bring it to the main sets of our adaptive immune response for inspection, right? Do you recognize this specific antigen? They usually, as I says, develop from monocytes.
So the T cells, they're on the lymphatic system, they're in the blood.
Well, they travel there through the blood. All right, so we asked the question, the T cells are only in the lymphoid system? No, T cells and B cells you'll find in your blood. But unless you do a special stain, you're not going to be able to differentiate them. You'll just see a lymphocyte, right? When they do differentials, that staining procedure to be able to differentiate the different white blood cells, they just see a lymphocyte. They don't know if it's a B cell or a T cell or a natural killer cell. Okay, unless you use a special procedure, and we have special procedures out there now where they can actually delineate the two different types of T cells. And this is particularly important for HIV infection because HIV targets a specific subset of your T cells, your helper T cells, your CD4 helper T cells.
But you can see with the leukocytes, you can see.
Yeah, leukocyte it just means white blood cell.
But you can still, like you said,
you will still see lymphocytes in your blood. You will see a normal level in your blood. Under when infection is happening, you'll see increases sometimes. And again, that'll clue you in as to what type of infection is going on, what type of invader.
And usually T cells would be viral infection.
Yeah, T cells you're going to see an increase with viral infection. But again, a differential is not going to tell you it's a T cell population. It's just going to tell you lymphocytes have increased.
Identifying and counting T cells is an expensive process.
Very expensive.
So it's only used when it's absolutely necessary, like AIDS patients.
Yeah, HIV patients. Because they really need to know that specific information. Yeah. So yeah, it's pretty high tech stuff, but it's pretty cool that we can actually do that. So in your blood, when they do a standard differential, the staining procedure, they're just going to know it's a lymphocyte. They're not going to know if it's a B cell or a T cell. And the awesome thing that we'll investigate in more detail when we talk about the adaptive immune response is that T cells and B cells have receptors that are specific for specific antigens, right? So they recognize right down to E. coli 0157, that specific strain of E. coli. right? You could produce T cells and B cells that recognize to that level of detail.
And it's after destroying
Yeah.
104, right?
Yeah, yeah. After the information has been presented to them by a dendritic cell.
After exposure once, it recognizes it the second time.
Well, in the first time, you're going to recognize it,
It has a
but the
40 days.
response is going to be longer, right? And that's something we're going to talk about later. Okay, so these guys generally reside, and that's where they get their name, right? Lymphocytes in your lymphoid organs and your lymphoid tissues, right? And so we'll talk about what those other tissues and organs are when we get to adaptive, right? We'll get into the specifics of that. Now, your other lymphocyte that you would see is your natural killer cells. These guys, the distinctive difference here is they lack specificity, right? They don't recognize specific antigens, right? They need help from the different components of the immune system, like antibodies that are produced by our B cells, to be able to actually recognize something to that specific level. They're not doing it on their own, okay? And as their name implies, they pretty much kill the cells that they recognize as being something wrong with them, right, via the aid of our other components of our immune system. So functionality is really what we want to focus on with these guys, right? So our neutrophils, as I said, are our phagocytic cells, right? Our monocytes and macrophages are our main phagocytic cells, and our dendritic cells really collecting and presenting those antigens, right? Part of our adaptive immune response for B cells and T cells, right? We're going to get into their functionality very specifically later. So at this point, it just says adaptive, but as I said, B cells and T cells, we're going to talk about their specific ability to recognize antigen, and then the special abilities of the natural killer cells. As you can see, you're going to see eosinophils with inflammatory response, right? And sometimes with parasitic worms, certain parasites, you'll see an increase. Inflammatory responses too, you'll see basophils. Okay. Do I want to start this one? It's a good question. Yeah, we'll just do the first one. We're just going to talk about, so we have surface receptors, right? I've mentioned cytokines and adhesion molecules. You've kind of already seen adhesion molecules in play when we looked at inflammation, right? How they were able to move into the area. Surface receptors are really important for detection, right, and communication between cells. And the cytokines are our chemical message. So sometimes we refer to those surface receptors as our eyes and ears, right? This allows them to see and hear their environment, right? This is how they interact with their environment. Very similar to how we see and hear things. The voices, which would be caught by the receptors, are the cytokines. So these are the chemical messages that are sent out, right? And they're going to bind to the receptors, right? The ears, and cause that cell to do something in response to that signal, right? If I yelled fire, everyone would run out of the room, right? So these signals have a particular reaction for a particular cell, right? Just like, and like I said, we're not going to go into the detail that you go into A&P, like with hormones in the endocrine system, right? But to give you an example of one that most people are familiar with is that insulin produced by the beta cells in your pancreas are stimulated when you have sugar in your blood, right? And then it's going to travel through your blood and attach to receptors on your cells to tell them, hey, sugar is here. Which seems kind of backwards, right? Why couldn't they just figure out that sugar's there on their own? But they won't take it in without insulin. And so therefore they starve if they don't know that the sugar is there and available, okay? So same thing here. In order to do things, they need to be told, right? They need that signaling. They need that chemical message. And then sometimes they literally need to grab on and travel to places like we saw with inflammation. They almost need receptors that are specifically allow them as hands to grab on, right? And we clearly saw that in that process of inflammation, of getting those white blood cells to to grab on to the endothelial cells within the blood vessels and be able to travel into the tissues where they're needed. Those adhesion molecules, and they call things like selectins and integrins. We're just going to keep it at this basic level, right? Understanding the adhesion molecules are doing just that. They're adhering, right, and allowing them to stick. So surface receptors, these are membrane-bound proteins, right? This is why we have proteins in our membranes. These are our signaling molecules, right? The receptors are specific, right, to the molecules they're binding to. The general term given to molecules that bind to receptors are referred to as ligands, right? You may have heard this term before. They're the key that goes into the lock, which is the receptor, right? So these are specific for one another, right? Insulin only binds to insulin receptors on cells, okay? If it doesn't have a receptor, it's not going to bind, it's not going to cause something to happen. So the same thing happens, right, for any receptor system. These are specific, okay? There is a specific receptor for a specific molecule, a ligand that binds to it. And that's just to pass a message inside to the cell, right? It'll probably be, that information will probably carried all the way to the nucleus, the command center, right? And molecules will interact with the DNA and cause that cell to do something in response to that message. So cytokines is the general name we give to those chemical messages, those voices of the immune system. So there are several different classes that we talk about. There's a whole bunch of them that are classified as chemokines. Chemo refers to chemical, taxis, movement, right? So these are involved in chemotaxis, right? So there's one that we'll talk about in the complement system. That's what it's doing. It's calling the white blood cells in. That's what we call inflammation, right? It's an inflammatory cytokine as well, because it's causing the movement of specific white blood cells to the area. Colony stimulating factor, we already mentioned this, right? This is the group of voices, chemical messages that are going to be sent to the bone marrow and tell us we need neutrophils right now, right? Or we need all the granulocytes, or we need all the lymphocytes, or we just need a whole bunch of B cells, or T cells, or boy, do we need macrophages. Really amp up production of those guys, right, above normal levels. Interferon, as the name implies, it interferes. And this is a chemical message specific to viral infections. So this is released by cells that are infected by viruses, and it's going to cause other cells to respond in a way that will protect them against viral infection, or protect the spread, I should say, of viral infection. So we're going to look at that more specifically when we get to that particular response. Interleukins, oh boy, that's a book and a half, probably at this point. These are a particular group of cytokines that specifically talk between white blood cells, right? That's where they get their name, interleukin. Inter, in between, leukin referring to leukocyte, white blood cell. They're commonly abbreviated IL, right? So whenever you see IL, they're talking about an interleukin, and then they're numbered. Some of the most common ones, of course, are the first ones we discovered were like interleukin 1 and interleukin 2, but I think it goes up into the 40s now, and the numbers. And I'll show you some examples of those. By no means are we memorizing, like I said, individual ones. We're not going to that level of detail. Tumor necrosis factor, these of course are a very active area of research, this group of molecules that can instrument an inflammation when we have tumor cell formation. So even interferons, the different interferons have been used for treatment of different virus infections, right? Where we've produced this outside the body as a drug and be able to give it as a therapy, additional interferon, more than the body is producing to help fight in viral infections. So as I said, here's some example of those interleukins. And so notice that a macrophage could produce it, or even our epithelial cells. And it's going to activate T cells, which is one of the reasons why it's one of the more common ones you'll hear. Activate macrophages, and it can even induce fever. T cells, this one cracks me up. T cells produce it to cause T cells to proliferate. They basically talk to themselves even, right? You ever have someone who does that, talks to themselves, right? Our immune system talks to itself even. Yeah, you know, they help activate themselves. So, and then as I had said, we have tumor necrosis factors, right, that can be produced by macrophages and T cells and natural killer cells. And this is going to help promote inflammation. It's toxic to the tumor cells and helps regulate certain immune functions. So this is a really active area of research. I think one of the most fascinating things when I was deciding to go to graduate school and why I picked immunology is there's so much we don't know, right? In this class, we're just scratching the surface, but in general, we're just scratching the surface and all the communication and stuff that happens between these cells and why certain things happen and don't happen when you have certain disease conditions. So it's really kind of fascinating area of research and lots of potential, right, for really good stuff to come from it. So we're just going to stick to the general understanding of the general main categories and classes, right? We're not memorizing this table, right? But those of us that like specifics, right, to help us understand the generalities, that is there for us. So
Do we need to know anything except like the general basics?
just the basics. Know the general description of each one of those categories, what they're doing, and that they're all cytokine groupings. So I'm going to go over this last one, and then I want to hop over to see where we are in our case studies. So adhesion molecules, as the name implies, just helps them grab onto each other. Really important, as we saw in the recruitment of the white blood cells out of the blood moving into the tissues, right, when they're called there. That's those guys in a nutshell. And we've seen those in action already. So let me end that. So we'll pick up, continuing on that same PowerPoint. You'll notice we barely made it a scratch in it, it seems like. Oh, this one please don't do this.